especially welcome. I just want to say that we have another talk coming on November 3rd, and this is going to be about plants, personality and communication in plants. So that'll be really cool. Rich Carbon, who's from UC Davis. So that's the next one, and that will be in person. We think we'll even have food by then. Hopefully,
I think so.
I think
CPSC
finally
have food. So
okay, so there must be a way, right? So okay, we'll work on that. And I'm really happy to have Stacy here. Stacy was a grad student in our program long ago. When did you graduate? 2015 or something?
2014. So it's been 11 years.
Wow. And she's done many cool things. Was working on gorillas back then and now has gone on to do many other projects, which we'll hear a little bit about today. So welcome, and let's get started.
Great. Thank you, Susan. I really wish I could have been there in person today. I think maybe last time I was in that room was actually when I gave my back talk when I finished my dissertation. So it's been a while, and this feels weirdly appropriate. So today I'm going to be talking about some of the work that I have done on early life adversity and its effects on aging and resiliency. But for those of you that I do not know or have not had the opportunity to meet yet, the overarching question that motivates all the research I do is, what are the evolutionary causes and consequences of social relationships in primates? And it is true that I mostly study non-human primates, but I do dabble in humans as well. So I use the various kinds of data that you see here to try to understand things like the relationship between social organization, behavior, and physiology, the health and fitness consequences of relationships, and the impact that those relationships can have on life history trajectories and thus on kind of larger evolutionary dynamics. Now, for the last several years, one arm of my research has focused on the effects of early life adversity. Now, I think we all probably have a fairly good idea of what we mean when we say that. Many of us probably think of things like the Dutch hunger winter during World War II, or maybe the horrific floods that occurred in Pakistan during COVID. But we need to have a definition to be good scientists. So we're going to define it as experiences that represent a negative deviation from the expected environment. I think everybody can probably agree that examples like this clearly fit that definition, no matter where you grow up. But there are many things that are more mundane and sadly more common that can also be considered negative deviations from expectation. So some of you are probably familiar with the very long-running Adverse Childhood Experiences, or ACEs study, which defines various forms of abuse, neglect, and household dysfunction as sources of early life adversity in humans. Now, a big part of the reason that there's so much interest in this topic is because early adversity has a really wide range of consequences that in some cases can stretch across the entire lifespan. There's significant evidence for tolls on physical and mental health. So just for example, it increases your risk of developing heart disease, diabetes, and depression in adulthood. But for a long time, many people believed that this was in large part because of the behavioral effects of early life adversity, which is itself correlated with a variety of what we sometimes euphemistically call health-reducing behaviors. So things like smoking, being sedentary, drinking too much alcohol, etc. So basically, we believed that the physical and mental health effects were things that were largely behaviorally mediated. Unfortunately, we can turn to other species to test our assumptions about this behavioral mediation. So early adversity is absolutely not something that only happens to humans. Fish, birds, insects, other mammals, and even plants all experience their own versions of what would be considered a rough childhood for humans. So early life adversity has long-term consequences for other species too. So I'm going to give you a very incomplete list here. Things like shortened lifespans, impaired social relationships, poor health measured in a variety of ways, and altered reproductive and stress physiology. And if you're thinking to yourself that these things sound suspiciously like some of the effects that we see in humans, you would absolutely be right. And I would argue that that really calls into question to what degree the effects in humans are indeed behaviorally mediated. So things like finches and hyenas are obviously not smoking or eating too many Doritos. So a common thread here is that all of these impacts could have major, usually negative, biological fitness consequences for the organism in question. And that means that understanding how early life adversity shapes these outcomes is crucial for understanding the variation in life histories and ultimately for evolutionary dynamics. I mean, in the evolutionary social sciences, there are two basic classes of hypotheses that are very commonly invoked to explain the relationship between early experiences and later life outcomes. The first, which I will kind of broadly call predictive models, emphasize selection for organisms to use the cues that they receive during development to alter their physiology or morphology in ways that might be beneficial to them in their future adult environment. Now, the other class of models are something that we'll broadly call constraints models that emphasize the immediate benefits of the phenotypic adjustments that organisms make in poor developmental environments, but also emphasize the long-term costs of those adjustments. Oh, sorry, we've got an animation problem there. So if you're somebody who follows this literature very closely, then over the course of my talk today, you're going to see data that are implicitly testing either one or both, actually, of these two constraints models in particular, the very broad developmental constraints model, and then also the biological embedding hypothesis. However, for the sake of simplicity and in the interest of not getting bogged down in details that, at least in theory, differentiate those very long lists of hypotheses, I want to frame today's talk as covering three questions. So the first question is, what causal pathways might help us explain the connections between early life experiences and adult outcomes? The second question is, do we observe interspecies variation in response to early adversity? And if so, why might that be? And for the third, I actually want to introduce you to a project that is currently in the works. And the focus here is on trying to expand essentially the comparative portfolio of early life effects research. So let's start with this one. How do the connections between early experience and adult outcomes happen? And for this analysis, we're going to be turning to the savannah baboons that are monitored by the Amboseli Baboon Research Project. I know that this is a project that probably many of you are already familiar with, but in case not, this was started in 1971 by Doctors Jean and Stuart Altman in the Amboseli region of southern Kenya, which you can see pointed out by that big red dot there on the map. And when I started working with the Amboseli team in 2019, it was specifically in order to address questions about the impacts of early adversity. The baboons are exceptionally good subjects for this topic. And the reason for that is because savannah baboons live in a highly variable environment and are very social creatures. And that combination means that they can grow up with no adversity whatsoever, or they can grow up with a whole bunch of different kinds of socioecological hardships. The Amboseli team has defined 6 different sources of early adversity for their baboons. They include being born during a drought, having your mother die or disappear before you hit 4 years of age, which is right before the onset of reproductive maturity for females, being born in a particularly large social group, which of course leads to a lot of resource competition, being born to a low-ranking mom. So baboons are very hierarchical, and life at the bottom of the hierarchy is really different than life at the top. Being born to a mom who herself is socially isolated, and finally, having a younger sibling that is born very close to you in time. And what that means is that maternal resources might be withdrawn before you would expect them to be. Now, we know that experiencing these things when you're a young baboon is bad for you, and that experiencing more of them is worse than experiencing fewer. So this survival curve that you see here has age on the x-axis, and on the y-axis, we have the proportion of subjects who are still alive at a given age. So baboons who experienced none of the sources of early adversity on this list are in dark blue. Animals who experienced any one of them are in light blue, any 2 are in yellow, and any 3 or more are in red. Now, hopefully, what immediately jumps out here is that we have a really big gap between the dark blue line, so those are our luckiest baboons, and the red line, which is the poor baboons who had lots of sources of adversity. So at any given age, each additional source of adversity nearly doubles a female baboon's chances of dying. By the age of 15, all of the animals in the 3 or more sources category are dead, which doesn't happen until nearly 30 for the animals who didn't experience any. So these are huge effect sizes. Now, besides knowing that early adversity predicts shorter lifespans, there are two other important background facts that I need you to know. The first is that elevated glucocorticoids predict lower survival probability in this population. Now, this is a massive oversimplification. I know there are people in the room who study glucocorticoids, but we broadly think of these as quote unquote stress hormones like cortisol, and excess exposure to them over long periods of time seems to be bad for mammals. So in the Ambacelli population, the animals represented by the blue line here have very low glucocorticoids relative to the rest of the population, and the animals in purple have very high GCs relative to most animals. Now, the animals in purple have a 38% greater likelihood of dying in any given year than the animals in blue do. The second important fact is that we also know that social connectedness predicts lifespan in the baboons. So females who are more connected to other females live longer than animals who have weaker social connections. The blue line is the females who are in the 75th percentile of social connectedness. The red line is animals in the 25th percentile. And we have a roughly 2-year gap in life expectancy at any given age. Now, all of these pieces of information led us to the conjecture that perhaps one way early adversity causes shorter lifespans is via increased glucocorticoid production. But also, if early adversity is keeping baboons from forming and maintaining strong social bonds in adulthood, then that itself might be a contributor to high glucocorticoids. So there are a variety of studies showing that relationships have health benefits, including lower stress hormone levels. So our conjecture here is that early adversity causes higher glucocorticoids in adulthood, but that this relationship is at least partially mediated by the strength of the animal's social bonds once they reach adulthood. So in order to test this, we use data from 197 female baboons. We focus on females specifically because in this species, males disperse at sexual maturity, so the necessary longitudinal data for them is just a lot sparser. From these 197 females, we have just over 9,000 fecal samples that we use to measure glucocorticoids non-invasively. We also have social bond strength measures from across the lifespan. And then for each female, we had complete information on those same 6 sources of early life adversity that I described earlier. Now, today, I want to focus on the effects of cumulative adversity, i.e. the total number of things that a young baboon experienced, rather than the specifics of individual sources. Now, in order to test our hypothesis here, we used a mediation model for this analysis, which is structured a little bit differently than the typical kind of multivariate regression that many of you are probably very familiar with. So I want to briefly explain the premise. Mediation models estimate 3 different kinds of effects. The first one, called the direct effect, is just the direct relationship between our predictor and our outcome variable. The mediation effect is estimated based on 2 different relationships. So first, the one between the predictor and the mediating variable, and then also between the mediating variable and the outcome. And then what we refer to as the total effect is the mediation and direct effects put together. So you can think of this as kind of the global relationship between the predictors and the outcome. Now, in our case, the outcome was fecal glucocorticoid values. The proposed mediating variable was social bond strength. And of course, both of these are measured across adulthood. And the predictor that we're going to focus on is cumulative adversity. And this, obviously, we measured during development. Now, I'm going to specifically talk about results from 2 categorical models. The first is going to compare animals who experienced no adversity to the animals who experienced any one or more sources. And the second is going to compare animals who experienced one to those who experienced two or more. So, what did we find? First, I want to show you the total effect of cumulative adversity on fecal glucocorticoids before we break it down into its component parts. Indeed, we find a large total effect of early adversity on glucocorticoids. So on the x-axis in this plot, we have the percent difference in fecal glucocorticoids. The black dots, of course, represent the estimated effect size, and the lines around them are the 95% confidence interval. Now, in gray, we have effect sizes and confidence intervals associated with each individual source of adversity, which I've put up there just in case anybody's particularly interested in one of those. But again, what I want to draw your attention to are the cumulative adversity models. So in the model that compares females who experienced zero sources of adversity to those who experienced one or more, the higher adversity females had GCs that were roughly 9% higher. And in the one source versus 2+ model, the higher adversity females had GCs that were about 14% higher. So that's our total effect. But this does not answer the question of how that total effect breaks down between mediation versus direct impacts. So next, let's look at the first part of that mediation relationship, the connection between early adversity and social bond strength. And remember, our prediction here is that animals with more adversity are going to have weaker social bonds in adulthood. And again, this prediction was supported. So now, instead of having difference in GCs on the x-axis, we've got the difference in the strength of adult social bonds. So negative values, of course, mean weaker social bonds. The females who had no sources of adversity had weaker relationships than those who had only one. And females who had two or more had weaker relationships than those that only had one. Now, it's a little hard to know how to interpret the size of this effect because these numbers are kind of arbitrary when you're trying to map them onto the strengths of social bonds. So to give you a better idea here of what this looks like, the dotted line is a 1 standard deviation difference in social bond strength. So in this case, the females in our higher cumulative adversity categories had bonds that were somewhere between 1/4 to 1/3 of a standard deviation weaker than the lower adversity females in a given model. And next, let's look at the second part of that mediation effect. So this is the relationship between social bond strength and fecal glucocorticoids. Again, both of them being measured in adulthood. And we're proposing that stronger bonds should mean lower glucocorticoids. And indeed, this prediction was supported as well. So having social bonds that were 1 standard deviation greater predicted fecal glucocorticoid values that were about 6% lower. So those are the components of our mediation effect. What about the direct effect of cumulative adversity on GCs? And it turns out that this relationship is quite strong. In the 0 versus 1+ model, the higher adversity females had GCs that were roughly 8% higher, while in the 1 versus 2+ model, the higher adversity females had GCs that were about 13% higher. Now, what this means is that the direct effect accounts for nearly all of the relationship between these variables. This is the same figure as in the last slide, but with the size of the total effects plotted alongside the direct effects. The total effects are those closed circles and solid lines, and the direct effect is the open circles and dashed lines. And you'll see they look nearly identical. And what that means is that there's just simply not a lot left over for the mediation effect to explain. So in this version of the plot, we're looking at the size of the mediation effect with those little tiny triangles that are kind of butted right up against 0 there. And here I've highlighted them in red boxes. The direct effect, again, is the open circles, and the total effect is the closed circles. So the direct effect was 11 times stronger than the mediation effect in the model that compared 0 to 1 or more sources of adversity, and 23 times stronger in the model that compared 1 to 2 or more. So to recap, because we have a lot of moving pieces here, cumulative adversity has a strong overall effect on adult glucocorticoids, raising them somewhere between about 9 and 14%. Almost all of that was accounted for by the direct effect, which increased GCs somewhere between 8 and 13%. Animals with more adversity did indeed have weaker social bonds, but only by about a third of a standard deviation at the outside. And animals with stronger bonds also did have lower GCs, but a 1 standard deviation difference only means a 5.8% decrease in glucocorticoid values. So where does this leave us? In spite of the fact that all of our predictions were supported, we found very little evidence that social bonds are mediating the relationship between the early adversity glucocorticoid connection. So the good news here from the baboon's perspective is that while early adversity does seem to compromise social bond strength, it only does so a little bit. But the bad news is that even quite large investments in social bonds seem to have relatively small effects on glucocorticoids. And what that means from the perspective of the animal is that it really doesn't seem like it would be physically possible to sort of fix the negative effects of early adversity on glucocorticoids via your social relationships. Now, as I was working on this analysis, however, one of the things that I found myself wondering a lot about was the generalizability of these findings. So baboons are fascinating, but they have a very particular ecology. Their environment is highly seasonal, so they go through significant food shortages for parts of the year. They also have these very strong inherited female dominance hierarchies that, of course, can have big impacts on resource access. And an important limitation is that we have almost no information about what happens to males because baboons' sex bias dispersal means that we just simply don't have the necessary longitudinal data on individual males. And that brings me to the second question I want to talk about today. Is there interspecies variation in responses to early adversity? And if so, why is that? Now, for this, we are going to turn to the species that most of the UCLA people associate me with, mountain gorillas. This is still the non-human primate species that I study the most. I work at the Karasoke Research Center in Volcanoes National Park in Rwanda. So Karasoke's origins also go back a long time to 1967, when Louis Leakey hired Diane Fossey to start the project that eventually became Karasoke. So Dr. Fossey habituated several social groups of gorillas, and they and their descendants have now been monitored almost continuously for about 60 years. And as you can probably tell from these pictures, this is something that has grown into a very large project over the years. So the research center itself is in Rwanda, but Volcanoes National Park spans the borders of Uganda and Democratic Republic of Congo. There are somewhere between about 650 and 700 mountain gorillas that live in the park across those 3 countries, which is more than half of the world's total population of this highly endangered great ape. Now, mountain gorillas make for a very nice contrast with the baboons. They have a very different ecology, which you can probably see just from looking at this picture. They are often described as living in a salad bowl, and there's a good reason for that. So unlike the baboons, they also do not inherit their dominance ranks, and they also have this interesting flexible dispersal system that means that it's possible to follow both sexes all the way across the lifespan. And of course, being a great ape, their genetics and their life histories are both meaningfully more similar to humans than that of a baboon. Now, before we could do anything with gorilla data that was as complicated as what we had done with the baboon analysis, we needed to answer, frankly, a much simpler question. Do mountain gorillas, like baboons, have shorter lifespans if they face more early adversity? We were basically starting from zero with the gorillas since nobody had studied this topic in them before. And I want to take a moment to acknowledge that everything that you see in this next section was led by Dr. Robin Morrison, a fantastic former postdoc who is now a group leader at the University of Zurich. So for this, we used data on 164 gorillas who had been monitored from birth to at least 6 years of age, and they were divided almost equally between males and females. And we deliberately tried to keep the sources of adversity as comparable as possible to the ones that were used for baboons. But of course, anytime you've got a species with a different socioecology, some things are just not going to directly translate. So for gorillas, we ended up with the list of adversities you see here, which basically we based on our collective knowledge of gorilla behavior, life histories, and socioecology. So we've got death or disappearance of your mother before you're age 6, having a competing younger sibling, so that one is directly comparable to one of the Ambicelli sources. The death or disappearance of an animal's social father, which we define as the male that was dominant during their first year. Experiencing group instability, so basically a group fission or disintegration. Infanticide, which means experiencing the infanticide of a group member who is close in age to you when you're young. And then being raised in a group with few age mates, which we defined as a mean of fewer than 2 age mates in their group when they're young, which for gorillas is kind of a form of social isolation, essentially, given their typical rearing patterns. So, does early adversity predict shorter lifespans in gorillas? I will fully admit that I was quite surprised when the answer turned out to be no, it does not. So, on this survival curve, the animals with the most adversity are in dark purple and the least in light green. And notice the positions of these lines are actually flipped exactly backwards from what they were with the baboons. So the animals with the most adversity are actually living longer. Experiencing 3 or more sources of adversity was associated with a 70% reduction in the risk of death across adulthood relative to experiencing none at all. Now, once we dug into this somewhat surprising finding a little bit more, it became clear that the effects were sex specific. So for females who are represented in the right-hand graph there was just no relationship between adversity and lifespan. It was for the males specifically for whom having the most sources of adversity was associated with especially long lifespans. But what's going on here, right? This finding, like I said, took us somewhat by surprise. There are 2 potential explanations. The first one is something called viability selection. So the idea here is that maybe early adversity is killing off weaker gorillas, and so only the strongest ones who have the longest lifespans, because they are particularly high quality animals, they are the ones that survive. Now, the pattern that we see in the male data is consistent with this idea. I don't think any of us think that it's very plausible that early adversity actually lengthens their lifespan. Instead, it's probably the case that if you are a male gorilla who managed to survive 3 or more of those things, then you're just a particularly tough animal. But I would also argue that viability selection probably does not account for all of what's occurring. For one thing, we see no evidence of the more adversity, longer lifespan pattern in females. And it's also the case that patterns of death from maternal loss specifically are not consistent with viability selection. So maternal loss is obviously a really catastrophic thing for a young gorilla to face. And we know from our many decades of data that if you're a young gorilla and you lose your mom before you are age 2.4, there's a 100% mortality rate. Right? You're just dead, presumably because you can't survive without milk. But interestingly, after that, we actually have a 0% mortality rate. And if it were really the case that some animals are just tougher than others, then what you'd expect is to see a lot more of a gradient instead of this really hard cutoff at a specific age that is almost certainly associated with simply the physiology of your digestive system. So what's the second potential explanation? This one is a little bit more cheerful, frankly, and that is resiliency. So maybe gorillas are just better at overcoming early adversity than other primate species. That might be what the female pattern would suggest. And of course, that begs the question, why, right? Why would it be different in this species? One possibility is very simple. Of course, these animals live in a much more resource-rich environment than the baboons, and they're not typically kind of struggling to find food or water in the same way. But it also could be the case that the various specifics of their dominance mechanism and their dispersal patterns matter. Gorilla society is, sounds funny to say this, but essentially more of a meritocracy than baboon society in the sense that you don't inherit your rank And both sexes have a lot of social options across the course of their lifespan because they have very flexible dispersal. Now, we are actively working on exploring these things, but we do have some published data that suggests that social support in particular might play a role for the gorillas. So we'll turn again specifically to maternal loss, which again, it's a particularly drastic source of early adversity for a young gorilla. Now, before young animals lose their mom, they spend an average of about 13% of their time in affiliative contact with her and 39% of their time within a 2-meter distance of their mom, right? So they spend a lot of time with her, unsurprisingly. After they lose their mom, for the animals that become orphans, there is a 28% increase in affiliative contact with other group members and a 42% increase in proximity to other group members. So while they do have a net decrease in contact, they actually have a net increase in proximity. It is clearly not the case that these animals are just becoming socially isolated after they lose their mother, who is unquestionably their most important social partner at this stage of their lives. It's quite interesting to see who exactly is responsible for this. So there are specific demographic categories of animals that are essentially filling in that social hole that mom left behind. The gray dots here are non-orphans, and the black are similar aged orphans, whose time with subadult and juvenile females in particular jumps dramatically after they become orphans. Now, the other category of animals where their relationship strength really increases is with adult males, and in particular, the dominant male, who is also statistically the orphan's most likely father. So it seems possible here that the very tightly knit group structure of mountain gorillas might help these orphaned animals get through the challenges of losing their mom. So in mountain gorillas, so far, we have a very different story than we do for the baboons. We don't have any evidence of negative long-term survival effects, though it is very important to note that we don't yet know whether there are other potential health consequences. That is something that we are actually actively working on as part of a comparative NIH grant, where the goal is to understand why we have significant longevity differences in wild chimpanzees and gorillas. But we do think that we have at least tentative evidence here for the idea that social buffering is a potential resiliency mechanism in gorillas. But it is almost certainly the case that there are multiple reasons for the species differences, right? Believing that it would be monocausal seems quite naive about the basic way that biology works. And so I would argue that we're really kind of at the start of understanding what is going on here. Now, that said, it does seem like the gorillas are potentially a promising model for understanding resiliency to early adversity in primates, since their data do suggest that they maybe don't follow what we think of, at least right now, as the typical primate pattern. And that brings me to the final question that I want to talk about today. So how is it that we can expand the scope of comparative research in this area? And as I mentioned before, this is a preview of kind of ongoing and future work. So this project really started with a paper that I published a couple of years ago, in which Chris Kuzawa and I outlined the various ways that great apes could be particularly useful for early life effects research. That paper ended up leading to a conversation with a neurobiologist at the University of Pittsburgh, Dr. Stacy Rizzo, who studies cognitive decline generally and Alzheimer's disease specifically. Early life adversity has molecular consequences for organisms that essentially mirror the consequences of aging. And there's a very rapidly growing body of evidence that the cognitive decline that happens to some people as they age can have developmental roots. Now, most research on cognitive decline occurs in species like the ones you see here. So things like rodents, flatworms, and fruit flies, unsurprisingly, totally dominate. And when this research does occur with primates, it's almost always with rhesus macaques or marmosets. And while they are certainly much more closely related to humans than Drosophila, Even the most closely related of those is a pretty distant human relative with pretty big differences in genetics, life history, social organization, etc. So thus far, for pretty obvious reasons, frankly, great apes are almost totally absent. However, they would be amazing research subjects if we could figure out how to unlock their potential, because they might be able to provide better insights into the factors that mediate susceptibility to and resilience against cognitive decline. So, if aging is a product of genetics and environment, then the fact that we have access to populations of both captive and wild apes offers kind of an amazing opportunity. So, virtually everything about the life of a captive ape is controlled by humans, while, of course, wild apes are living in relatively unaltered environments. They're foraging for their own food. They have total control over their own movement and social partner choices, and obviously, sometimes to their detriment, no veterinary care. So there are lots of factors that could potentially change with age in both captive and wild populations, including social behavior, nest building, tool use, diet, locomotion, and then the one that we have chosen to focus on at the moment, which is brain pathology and biomarkers. So in humans, we can draw blood and measure a variety of biomarkers that predict cognitive decline, neurodegeneration, and Alzheimer's disease many years before anybody ever reports symptoms. So it gets super repetitive to say all those words. So I'm just going to use cognitive decline as a shorthand here. Now, on the left here, we have a list of some of these biomarkers. And collectively, they give us information about amyloid plaques, neurofibrillary tangles, and then white matter rarefaction. So amyloid plaques are abnormal protein deposits in the brain. Neurofibrillary tangles are bundles of tau proteins in particular that accumulate inside of neurons. And white matter rarefaction is the loss or the thinning of white matter. And as you can probably imagine, none of these things are good, right? Collectively, these are brain pathologies, and they're detectable in cerebrospinal fluid, blood, or both, well before you get any kind of actual cognitive impairment in humans. We also know that we can detect these biomarkers using the exact same assays in marmosets. So they increase in concentration with age, just like they do in humans. On the y-axis of these graphs, we've got the plasma concentrations of neurofilament light and beta amyloid 42/40 ratio. And on the x-axis, we've got age categories for the marmosets. So concentrations do change with age in all marmosets. But animals who specifically have a presenilin-1 gene mutation that's associated with early onset Alzheimer's in humans start developing these indicators much earlier than the control marmosets do. So they're represented here by the blue dots, whereas the control animals are in black. Now, we also know, thanks to some work that was published quite recently, that we can measure at least some of these biomarkers in fecal material in humans. And we can do this because some of these markers, interestingly, are actually expressed in the gut. There's a lot of interest in the role that the gut-brain axis is playing in the development of cognitive decline. But even for the biomarkers that aren't actually expressed there, these are soluble proteins that move across the blood-brain barrier and ultimately will end up in the gut. So patients with Alzheimer's disease have higher concentrations of amyloid beta aggregates in fecal samples than healthy control patients do. And finally, we do know that at least some great apes can suffer some of the same age-related brain pathologies as humans. Now, this has been widely reported in the scientific literature, but it's remarkable how anecdotal it remains. So it is not at all clear how common these are, at what age these pathologies begin developing, or what the health or behavioral consequences are. And of course, we also currently have no ability to detect any pathology pre-mortem because you can't routinely collect blood from great apes. Which brings me to the new project that I'm working on. So Dr. Rizzo and I are working on determining if it's possible to non-invasively measure cognitive decline biomarkers in great apes. We are working with a bunch of different zoos around the country to collect matched fecal and plasma samples from adult chimps, bonobos, gorillas, and orangutans. I apologize if you can hear a bunch of music in the background. There is a motorcycle parked outside my window blasting music. Hopefully they'll move on quickly. So our goal here for this project is to figure out if we get comparable results in plasma and in feces. And if so, this would be really cool because it would open the door to all kinds of longitudinal monitoring that is currently impossible, as well as allow us to answer some really basic questions like which pathologies do they get, how often, and at what age are they starting? So if this is successful, we would like to expand the project to include wild apes and then integrate behavioral data collection to try to determine if these pathologies are actually leading to cognitive impairment in the same way that they do in humans. That's currently an open question. If you talk to many zookeepers, they will anecdotally report that they see things in older apes that they believe might be indicative of cognitive decline, but actual analyzable data are still essentially totally absent. So we've currently got approval from something like 18 zoos that are collecting data, and our proposal is pending at about 30 more. So we're hoping for a pretty decent sample size here. Even though I don't have data to share yet, I did want to talk about this project. I'm very excited about it because in some respects, I feel like this is sort of the tip of the iceberg. So great ape research has historically been very bimodal, right? We have completely non-invasive fieldwork that strictly relies on observational data. This kind of data has great ecological realism, but we also really are limited in our ability to measure lots of parameters of interest for many questions. And on the other hand, we've got very specific instances of great apes being used in biomedical research, probably, most famously for HIV and AIDS. This kind of work can obviously make really important scientific contributions, but it doesn't really have any ecological validity, and it also, of course, presents some serious ethical concerns. Now, the good news is that I would argue that advances in technology and the increasingly impressive number of data years that we have available means that this gap is something that is increasingly bridgeable. So this map is from that 2023 paper I mentioned before that ended up leading to this project. It shows all of the great ape field sites in Africa. First, there's a lot of them. But second, I want to point out the timelines. So anything that's represented on this map by a circle is a project that has been running for 40 or more years. And the upside down triangles are projects that have a 30 to 40 year history. That's quite impressive, represents an awful lot of work, but it's also really scientifically important because it means that we've got information on multiple generations of great apes. So we always have a problem with great ape research with small numbers and slow life histories, but these are becoming less and less of a problem over time, thanks to the many hundreds of years of collective work that people have invested in these sites. I don't think I probably need to sell this point very hard for the Beck audience, but great apes are obviously a better analog for human health and behavior research, even compared to other primates. They are genetically, behaviorally, cognitively a lot more like us than even a rhesus macaque is. And their life histories are a lot closer to ours on a variety of dimensions. Now, it's also the case that I think that something that has been kind of underappreciated is the amount of within clade variability that we've got. So across the various genera, we actually have a remarkable amount of variation in ecology, in diets, behavior and cognition, their social organization, the degree of sociality that they express, and even their life history characteristics. So I think we can hopefully leverage this variability in all kinds of different ways, certainly in early life effects research, but also for lots of other purposes. Well, great apes really have been an important part of the bioanth tradition for a long time, and they certainly are an important source of basic science insights on things like the uniqueness of our species and the way that environments influence organisms like us. But I like to think that research on them also has the potential for real-world impacts. So my collaborator, Stacy Rizzo, once said to me, if mouse Alzheimer's were the problem we were trying to solve, we could have cured it 50 times over by now, right? The point she was trying to make is that we really need to be trying new things. And I think with some creativity, great apes can potentially be part of that story. Also, attractively, Figuring out secrets to healthy aging and resiliency, yes, of course, it would benefit humans, but it also helps benefit the animals themselves. Right? There are thousands of these animals in human care, and interventions that could help us can also potentially help them. Now, before we go to the Q&A today, I want to leave you with just a few last thoughts and questions, frankly, about the early life effects story. So first, I think it's safe to argue that there's a lot of emerging evidence, and not only from primates, that there's a considerable amount of within and between species variation in response to early life experiences. So thus far, I'm not sure that we've done a very good job of using that variation to our advantage because we didn't have a very good grasp on the range of responses that exist. Now, as that changes, I hope we're going to start to see more progress on understanding mechanisms in particular. Second, and this is something I discussed with some of the graduate students I met with one-on-one earlier today, I've become increasingly convinced that this is a literature that is really suffering from a pretty significant file drawer problem. So all literatures do to some extent, but as somebody who has spent a lot of the last 5 years publishing null results in this area, this is what I showed you today is absolutely not the extent of the null results that I've published. It's kind of discouraging, frankly, to see how much pushback there is from both reviewers and editors when the results don't conform to these very strong expectations that people have about the effects of early adversity. But the upshot of that is that the story might not be nearly as bleak as we think it is. And so that's kind of the cheerful part of that unfortunate reality. And then finally, I think that there are really important questions about this, whether this literature is doing a good job of making the right comparisons. So a lot of it focuses on comparing organisms who experienced early life adversity to those who didn't. And that is built on the assumption that early life adversity, early life specifically, is particularly catastrophic. And that might very well be true, but properly testing that assumption means that we need to do a lot better job of measuring the effects of adversity at other life history stages, right? Not only what organisms experienced during development. So aging is fundamentally something that is a cumulative process, and development is only one piece of that cumulative whole. So with that, I want to thank the many collaborators and co-authors who've contributed to all this research in a whole lot of different ways. Thank you to Susan Perry and the Beck organizers for inviting me today, and all of you for listening. I'd be happy to take questions.
All right. Can we take questions from Beck students first?
Thank you for that talk. Really liked it. That was great. So in your last few slides, you talked about other points of life history or other points of development being important to study for points of adversity. Would you give an example of what a potential point in development might be in an area that is currently under study?
Yeah. So basically, okay?
Yes, I could hear that just fine. Thank you. Sorry, 1 second. I have to partially close this blind because I'm being blinded by the sun. Sorry about that. If I kept moving around, that was why. That's a great question. And essentially what I mean by that is anything that comes at sexual maturity or later. So there's a huge focus on adversity before you hit sexual maturity, right? Sometimes in utero, during infancy, there's controversy over to what degree other primate species have childhoods, right? However you want to define that, the juvenile period. But then after that, it kind of tends to disappear. And I'm not necessarily saying that I believe that all parts of life are equally important. I think that seems probably naive. But when we are doing something where we're just comparing something that happened in development to you versus something that didn't happen to somebody else during development, we're ignoring what the potential effects are of things that are going on later. And I don't think literature has done a very good job of essentially just acknowledging the reality that we really should be doing both to try to figure out whether this represents as much of a sensitive period as we think it does, right? One of the other kind of interesting I don't even know what the right word is, but paradoxes is that there's a whole other part of the literature that talks about organisms during development as being especially plastic and especially resilient. And so we've got this weird situation where we've kind of got multiple stories being told in different parts of the literature. And I think looking at adversity, the effects of adversity at and after reproductive maturity would help us disentangle some of those things.
Thank you. Really appreciated it.
Thank you. It's a wonderful talk. I just was wondering, I think I missed, how were you measuring social bond strength in that first baboon study?
In the baboon study, yeah, that is measured based on grooming patterns. This is terrible. It's been too long since I've written that paper to tell you exactly how we calculated it. I can't remember, but basically it's a measure of how social, what is the strength of your grooming bonds with the top 3 females in your kind of social network relative to others.
Abby. Nice to see you. My question kind of comes off of the question that was asked. So I'm curious. I think I have one question to ask first before I ask my real question, which is, were social bonds measured after maturity only?
So yes, if for purposes of this study, all of the social bond measurements that occurred as part of the mediation variable, all of that is after animals hit sexual maturity. Yep.
So then that into my real question, which is, I know from studies in human cohort where folks that are in orphanages who face early life adversity or in orphans, and then also foster care systems, looking at social bonds in those contexts, it seems like the data support an idea that early life adversity has stronger effects biologically when social bonds during early life are less. And considering for for the baboon data in particular, you measured social bonds in after sexual maturity, but potentially there could be an effect, and I want to know what your thoughts on that, about if social bonds in early life when adversity is experienced potentially could be a mediating factor in the impact of on later life health. So measuring the bonds at a different time than
yes, I think that's a fantastic question, and I completely agree with your read of the human literature and what it suggests about sort of a sensitive period, for lack of a better way to put it. Unfortunately, the reason, or at least my understanding, and I would definitely want to defer to the Amboseli Baboon project directors here because they collect a lot of data. But my understanding is that we, just because of the way data are collected, we don't have data on independent social bonds from animals before they hit sexual maturity. Just because of the differences in their patterns of interaction. Essentially, you're not comparing like, and the project has to make strategic decisions about what they do and don't collect. And so my understanding is that we've got a data limitation that means that we couldn't do that in the same way that we can do with adult bonds. But again, I would want to defer to the Amboseli directors there because I'm not 100% sure about that.
Thank you.
Thank you so much for this presentation. It was really cool to hear all these experiments that are going on. I was really interested in the gorilla experiment and how like females, there was not really an effect, but for males, they live longer. And I was wanting to see any connections between the way gorillas build relationships and then how humans build community resilience.
So I'm just to ask a clarification question. I'm trying to connect the first part of your question to your second, because I'm having a little bit of trouble here. So if I understood correctly, you were asking about the sex differences that we see between male and female gorillas. But then the second part of that was about resilience. But can you repeat exactly what it was?
Can you say that one more time? Sorry.
Can you hear me? I'm getting
Yes,
a lot
I can
of
hear you now.
Okay. Sorry, I'm getting a lot of feedback for some reason. So can you just repeat the second part of your question? So the first part was about the differences between male and female gorillas, right?
I was more interested in the male side and the kind of emphasis on resiliency with there. Well, there's two explanations, but on the resiliency end, do you see like any connections with maybe how they build relationships and what that looks like for their resiliency, and how community or how like humans build that same like community resilience, if any connection at all?
Yeah, that's an interesting question. So I think, just to be very clear, the males are specifically the sex in which we do feel like we have at least some evidence for viability selection rather than resiliency. Now, that said, it is important to note that those two things do not have to be mutually exclusive, right? It actually is possible that both things could be occurring simultaneously. I do think that one thing that is interesting about the gorillas, especially relative to the baboons, is male gorillas in particular can lead, certainly not all of them do, but many of them can essentially be solitary for most of their adult lives. And for lack of a better way to put this, they seem to be very good at dealing with not having a lot of strong social bonds, right? That's just sort of part of the life history of many, many male gorillas. And so I'm not sure that I'm not sure that I would necessarily predict that matters to them in the same way that it does to a female gorilla, or certainly a female baboon, where they have these extremely strong social relationships, in particular with other female relatives. How that maps onto humans, that's a really hard question. I think there's always really complicated questions to be asked about the degree to which these animals' lives can kind of be mapped onto human conceptions of things like resiliency. And I guess I'm not sure that I'm entirely comfortable making that leap. Which I fully recognize that seems like a total cop-out, but.
Thank you.
There's also a question on
the chat.
Oh, here.
That says any autism over time affects various organs. Can you hear me?
That's a good question. I don't know if anybody has looked at that. That's a really good question, and I don't know the answer. Frequently, so one issue we do have, baboons very often just disappear. It is relatively uncommon to be able to recover the bodies. For gorillas, we often are able to recover bodies, certainly not 100% of the time, but it usually is the case that there's a decent time lag between when the animal dies and when we're able to recover it. And what you see is a fair amount of organ degradation because of course, organs degrade pretty quickly. And I feel like that would limit our ability to say you probably would need essentially higher quality necropsy data in order to be able to say anything about that with any certainty. One of the limitations of field studies, unfortunately.
Dan?
Hi, Stacy. Wonderful to see you.
Hey, Dan. Good to see you too.
Unsurprising. It's not at all surprising to see you doing great work. So I have a couple of questions, comments. The first, so in the baboon data, you pretty clearly ruled out any significant behavioral mediation, but the sample sizes were quite large there compared to the gorillas. And I mean, the gorilla sample sizes are large impressively for gorillas, given how few animals there are, how small their social groups are. But still, if we then go and split that by sex, the numbers get smaller, right? So curious what you think of the conjecture that the unexpected result in the male gorillas is a sample size effect, where in particular, if behavioral mediation applies in the case of the gorillas, right? If you have, say, what we see in humans, for example, ACEs leading to increased risk-taking in adulthood in a variety of different domains. If you have a smaller sample of gorillas to start with, a smaller sample still of male gorillas, and those who experienced adverse early experiences roll the dice and happen to succeed, maybe achieving alpha status and so on, and their GCEs go down, and as a consequence, their high rank. Then if you had a bigger sample, you see that a lot of them roll the dice and lose, and you get the expected effect.
Yeah, I think that's a great question. So we unsurprisingly faced a lot of questions about the sample size when we were working on the gorilla analysis, because of course, as you pointed out, we're never going to get, we're never able to compete with Ampicelli. We don't have those kind of numbers simply because of the species. That said, if you work on the assumption that the effect size, and I fully realize the problems with this assumption, but if the effect size in gorillas were equivalent to what is in baboons, then we did a whole bunch of a whole bunch of power analyses, and basically we would have had a 99.999% chance of detecting the difference. Now, that doesn't directly speak to the problem that you were highlighting, and I'm not sure that we have a great way to disambiguate that. I mean, one of the things that I really would love to do and fully plan to do is rerun this analysis in 20 years, when we do have a bigger sample, because I think that there is a lot of stuff here that is really hard to disentangle. And you're absolutely right that as soon as we start chopping it up into these smaller blocks, we can become less and less confident about the conclusions that we're drawing. I mean, I think the sex specific thing in the gorillas is really interesting. And male gorillas and female gorillas have these really major life history differences because of what I mentioned earlier about, the male's propensity to go be, many of them go and be solitary for part of their lives or their entire lives. They do a lot of risk taking. There's a tremendous amount of risk taking that male gorillas are doing across the course of their lives that females just simply don't do. And frankly, I don't think that we have a good way at this point to disambiguate all those things. But what I can tell you is if we saw a pattern that was equivalent to what was happening in the baboons, regardless of sex, we are very confident we would be able to detect that.
Yeah, okay. That's a satisfying response, given the constraints that you're operating under. Let me just a little out of left field in another direction. So I totally buy why gorillas are a better comparison point than baboons, and why baboons are better than mice, and why mice are better than flatworms if we're studying humans. But there is a case to be made for looking under the lamppost, right? That is a species where we have a lot of accessible information, and maybe the phylogenetic distance is intermediary between flatworms and gorillas. I don't know if you can see them, but I have my dogs with me right now, and domestic dogs, I think, are an interesting model. One, their cognition has been studied pretty extensively right now, right? Two, it's relatively easy to get biosamples from to the free-ranging gorillas that you're studying, and three, there is, while there be gaps in the individual histories, there probably is substantial variation across readily available animals in terms of adverse early experiences, because owners who adopt rescue dogs will know the age of the dog at the time of adoption is estimated by a vet, and the physical condition of the dog at the time that it was rescued, right? And so the difference between a young dog that's starved and a privileged dog that comes from a breeder and was hand fed is going to be pretty significant. So, granted, the phylogenetic distances are than any of the primates that you're looking at, but the availability of the data is probably also.
Yes. Sorry, the microphone cut out there at the very end, so I apologize if I interrupted. So absolutely. And one of the things that I should emphasize is I view this as an and, not an instead of thing. I mean, I think, as I mentioned in one of those slides, is one of the things that is really sort of emerging is that there are very different effects across species. And I don't think that literature has done a very good job of leveraging that. And so in an ideal world, if we had unlimited funding, et cetera, of course, excuse me, we would probably be looking at this in a much wider range of species. Obviously, research money is limited, and so we do have to make strategic choices. One of the things that is very nice about the grade 8 populations that we do have access to is the data are remarkably detailed, and in many cases, we have essentially daily records from these animals from the day they were born, which is remarkable. That's thanks to the, like I said, collectively hundreds, probably thousands of years that we put in. And, one of the limitations, I feel like I didn't fully appreciate this until I started working with human data myself. The human data are amazing and you can get these really huge sample sizes in some cases, but you've also got very, very limited amounts of information about any particular person, right? And a lot of it relies on, self-report data, recall data about things that happened 20 years ago. Whereas for a lot of these great apes, both in captivity and in the wild, we've got written, complete information about everything that happened from the day they were born. And I think that we would be missing an opportunity if we didn't try to build on that. But that said, I don't disagree with anything you're saying.
Hi, thank you so much for coming and speaking to us. My question, I have kind of a couple questions integrated into one. Early life diversity selected for baboons. Specifically, could you kind of differentiate what defines a low-ranking mother versus the socially isolated? And then also, I guess, what is the feasibility of research in baboons, since it's pretty to research? And I guess, how would that be integrated if we added an ELA category of an absent father on baboon's life as well? So those are kind of questions integrated into.
Yeah, no, that's a really interesting question. So many people, I like your first question about the difference between social isolation versus included me when I started studying baboons. I just made the assumption that lower ranking females were more socially isolated. And it turns out that, at least at Ambacelli, that's not true. There are many low ranking females who have very strong social bonds with a lot of different females. But that doesn't mean that they're not at the bottom of the hierarchy. And there are also some very high ranking females that are quite socially isolated and really don't have much in the way of social bonds. So those two things are really quite well differentiated. And when we're talking about the dominance hierarchy in particular, what we're talking about is the outcomes of specific dominance conflicts, right? Essentially, who backs down when you are competing over resources. And baboons are a species in which this is something that's really easy to measure. First of all, they do a lot of competing, but they also have these really strict dominance hierarchies in a way that we don't have in a lot of species. So that's the relatively simple answer to the isolation versus dominance thing. I've always been curious about the paternal thing for the baboons in particular. I'm sure some of you know that, paternal care in the gorillas was what I studied for my dissertation. And so this is something that's always of interest for me. I will fully admit that I think that this has more biological salience for the gorillas than it does for the baboons, simply because young gorillas many of them have quite close social relationships with especially the dominant adult male in their group in a way that baboons do not with the males in their group. So keep in mind, these are always multi-male, multi-female baboon groups. There certainly is evidence that paternal care does seem to matter for specific types of resource access in baboons. There's fairly compelling evidence that they actually do know. They do have information about paternity that they use. But my suspicion is, given that the sort of distribution of the infant male relationships is not quite as acute as it is in gorillas, that it probably their quote unquote social father doesn't matter in the same way for a baboon as it does for a gorilla. That's my suspicion. But that said, it's an empirical question, and I actually think it would be really interesting to test.
Yeah, I was curious about, you're planning to look at the amylase levels in the feces of all these different species. I don't know anything about this, but are there different profiles, like thresholds of like acceptable versus unacceptable levels for each species? And like, would you create profiles of like these thresholds? Does brain maturity and brain size matter?
And honestly, the stuff you're asking about is part of the goal of the project. So currently, we just don't know anything. I mean, we are really, really starting from scratch. And that's the reason that it's very important that we have matched plasma and fecal samples, because it would totally be a leap to just go straight to the fecal samples. But one of the things that because it does seem like we've been very fortunate to get really positive responses from zoos about this, across all available great ape species, which is awesome. And we've actually requested samples from animals of all different ages. And so one of the things that we'd really like to do is essentially, as you said, build profiles and give zoos back basic data about who is it in your population where we see evidence of these markers, where don't we see it, what ages does it seem to be kicking in at, do we see interspecies differences? Frankly, all of that is totally unknown. Which in some respects seems surprising, but when you actually stop and think about what kind of data is required to get there, it's less surprising, right? We don't have a lot of grade 8 blood just kind of floating around available. And our project actually capitalizes on veterinary exams that are already happening. So it's sort of a slow burn project in the sense that we have to wait for animals to go through their routine veterinary exams, and then blood is collected at that time. But of course, that just relies on zoos, regularly scheduled procedures. And so it is quite slow. We essentially have samples trickling in over the course of 2 years.
Oh, that's super cool. Thank you.
Yeah, I'm looking forward to seeing it. So glad to hear other people think it's cool too. Any other questions? We do have 10 more minutes if people have more questions.
I assume Ashley already told. I can talk to you in my meeting with you. I won't ask questions if I'm the only one.
Oh, Ashley's got her hand up on Zoom.
Oh, good. Yeah, let's have Ashley talk.
I had a question about the gorilla study that you did when you looked at maternal loss for the orphan gorillas and the changes in their social relationships following loss. I might have missed this. I'm not sure if you mentioned it or if you had the data, but were you able to look at who was driving that change in relationship? Was it the orphan seeking out these contacts, or was it other members of the group who were stepping in?
Very good question. So for the males in particular, it is very much infant driven. So they are the ones who are initiating, and the males are essentially, they just become they're always pretty tolerant of kids, to put it very colloquially. But they become especially so when it's orphans. They will allow them to sleep in their nests. They'll allow them to kind of sit directly underneath them while they're feeding, which gives the infants access to, readily available food. In some cases, food that's a little bit easier to process because the male himself is like dropping pieces of thistles, nettles, things that are hard for infants to process because they go through a learning period. But that is very much infant. The initial content is very much infant treatment. For the subadult females and the juveniles, it is much more mixed. Those subadult and juvenile females show a lot of interest in the orphans, but the orphans also are perfectly happy to have interest expressed. Let's put it that way. So that is much more kind of mutual.
Very cool. Thank you.
Mm-hmm.
Any more? Does anyone else on the Zoom have questions?
Yeah, we'll let you have a from us before we're meeting individually then. Thank you.
Thank you, everybody. It's great to see you.